recording. And you'll see a little button pop up that is essentially your permission for us to record. So with that, I'll just say once again for the recording, my name is Beth Bisson. I'm the Associate Director of the Maine Sea Grant Program. Welcome everyone. This webinar on rockweed phenology research is jointly hosted by the Maine Sea Grant Program, New Hampshire Sea Grant Program, and Cooperative Extension programs from both Maine and New Hampshire, as well as the Schoodic Institute and Maine Maritime Academy. We have two guest researchers who will be presenting today. Jessica Muhlin from Maine Maritime Academy, who's a professor of marine biology, and Hannah Weber, Director of Marine Ecology at Schoodic Institute. Also with us today, we have the longtime co-manager of the Signs of the Seasons Phenology Monitoring Program, along with myself, Esperanza Stanchoff, Carrie Kaiser, who is the Environmental Literacy and Workforce Development Program Manager at Maine Sea Grant. If you just want to give a wave, Wells Costello, who is the Citizen Science Outreach Coordinator for New Hampshire Sea Grant, and Allison Eberhardt, who is a Coastal Ecosystem Specialist at New Hampshire Sea Grant. So hopefully I haven't forgotten anybody, and I know that we have many longtime volunteers with the Signs of the Seasons program and also New Hampshire's Coastal Research Volunteers program who have been personally involved in walking over slippery rocks and through tide pools to collect all the data that Hannah and Jesse will be sharing with us today. So I just want to extend a warm welcome and hearty thank you to all of you for being involved. And so with that, I will turn it over to Jesse to get us started.
Awesome. Thanks.
And I'm sorry to just interrupt you, Jesse, but I wanted to just remind everybody that live closed captioning is provided. And Carrie just put a link in the chat if that's something that you'd like to have pop up on your screen while you're listening. Sorry, go ahead, Jesse.
Great, thanks, Beth. So we've already given our introductions, and there's a lot of you on this webinar. So thank you all for spending your lunch hour with us. And if you want to share who you are in the chat too, I think that for some folks it might be nice to kind of just know who you are and where you're from. This webinar is hopefully going to help you understand why it's so important to monitor rockweed in a changing marine environment, and give you some insight in terms of what's happening with rockweed in this kind of New England, Maine area in terms of research and citizen science and the intersection of rockweed research, and then give you some insight in terms of what the data that the Signs of the Seasons and the coastal research volunteers have been collecting, and kind of give you a waypoint of what those data are showing right now. I think for a lot of you, you collected the data and you input it into Anecdata, and then don't kind of see the large scale patterns and trends that we might be seeing now. So we'll give you a little bit of that. And Hannah and I are going to kind of go back and forth in terms of both of us kind of giving little parts of this. And the other part of this objective of this webinar is to really celebrate all of you who have been steadfast volunteers in collecting data since 2014. And that it's really amazing. And as a researcher for rockweed, I really can't say how thankful and grateful I am to citizen scientists who are monitoring rockweed and going out and participating and collecting data and sharing it. Hopefully you've learned a little bit about rockweed as you've been collecting and appreciate it for just how amazing and awesome it is as an organism and as part of an ecosystem. So really wanting to thank you in this endeavor together. On the bottom of all of the slides, it kind of this kind of like timeline flow chart will kind of help you see where we're going. So if you're kind of figuring out like, oh, they're talking too long, or where are we in this? Hopefully this gives you a progression of where we are going. So to kick it off, in thinking about climate change in marine environments, climate change is occurring at the global scale and occurring in terms of thinking about not just terrestrial environments, but in the marine environment in lots of different physical and chemical changes. So in thinking about some of the physical changes that are taking place, changes in sea surface temperature, changes in ocean circulation, changes in meteorological precipitation and storm events, all of those things are occurring at different scales. So changes in variability annually, decadally, and on longer time scales, as well as changes in chemical changes. So thinking about increased carbon dioxide leading to ocean acidification, changes in the amount of oxygen in the near shore and deep ocean, and changes in the amount of nutrients that are available. So all of those are occurring, especially in the marine environment, and they're occurring quite rapidly within the last 2 decades or more. And in terms of kind of orienting you from just the large global marine environment to the Gulf of Maine where we are, there are marked changes in terms of thinking about one particular physical change, which is temperature. And so this particular data product that you're seeing right here is looking at the map of an ocean. You can see the latitude on the y-axis. And what you're seeing is a heat map in terms of looking at anomalies deviating kind of from the average temperature. And so the warmer the color, the hotter the color, the warmer the water is in terms of increased anomaly. So kind of shifting towards more warm conditions, and the cooler the color, the cooler those conditions are. And what you can see is this is the average temperature for 2021 globally in terms of climatological data. And you can see that the global ocean is getting warmer. So it's trending warmer. And in 2021, it was the 4th warmest year on record within the contiguous United States and 6th warmest globally. So water is getting warmer in marine environments. And if we just look at the Gulf of Maine, this is another kind of data product coming from the Gulf of Maine Research Institute, in which you're looking on the y-axis at the sea surface temperature anomaly. So again, kind of trending more towards the warmer conditions. And on the x-axis, it's over time. And what you can see is that the green dashed line is the global rate of increase in sea surface temperature over time. And the blue is the Gulf of Maine data set in particular. And so in general, the whole global ocean is warming, but the Gulf of Maine is warming faster than other global ocean bodies of water. And so we can see this in terms of thinking about just one particular physical characteristic that's changing with climate change in the marine environment. And so in thinking about just how warming temperatures might change in marine environments, what does that mean for the organisms that are living there? So temperature is a really important parameter for the health and well-being and the functioning of organisms and ecosystems. And so the biological response of climate change, and thinking about just one particular part of climate change in terms of sea surface temperature, is happening, and that there's a lot of information that's occurring and data being collected on what we're calling the briny deep. So in thinking about organisms and ecosystems that are offshore and in deep water. So climate change is occurring, that there is warming water, and it's affecting lobsters, both in terms of different parts of their life cycle and their life history, affecting cod, in terms of thinking about those being 2 very important commercial economic resources for New England, and also changing the phytoplankton, or the primary producers, the base of the food web. And so these rates of change due to climate change can have profound consequences on individual species, like let's just say a particular diatom or phytoplankton species, but also entire food webs. So changes in phytoplankton will have changes in the zooplankton or the calanus species that are here, which might have then implications in terms of thinking about the organisms such as right whales that eat those zooplankton species. And so that there are going to be these biological responses to climate change that we're seeing in marine environments. And those data are sparse, but they're accumulating over time in terms of we're just starting to really understand those responses at individual species levels and at ecosystem species levels. But the briny deep in the open ocean is a little bit different than what the intertidal looks like. So within the Gulf of Maine, we have a lot of intertidal ecosystems that span rocky shores, soft sediments, and salt marsh habitats. And so Hannah's going to tell us a little bit about responses to climate change in intertidal systems.
Sure, thanks, Jesse, and hi, everybody. Jesse, could you give me a thumbs up just so I can make sure you can hear me? Okay, thank you. Yeah, so if you wanted to just go to the next slide, that'd be great. So right, interesting, the briny shallow is a really resilient system. This is a system that has a lot of organisms that do the same thing. So there's a lot of like functional overlap in what things do. And so that makes the system super resilient to change. But Jesse mentioned all of these sort of pressures that are occurring in the briny deep, whether they're physical or chemical changes or the combination of them. And these, of course, are also happening in the briny shallow, and terrestrial pressures are also changing in the briny shallow. I don't know how many of you remember last summer in the Pacific Northwest, there was a heat wave, and this occurred at the same time that, oddly enough, low tides during that time were at midday, and millions of intertidal organisms were killed in a very short period of time. And so we're not sure what will happen in the briny shallow, in the intertidal with a changing climate, because this is a very resilient system. But then again, like last summer showed us, it's also a very, very sensitive system. We can say that we won't expect change to happen at the same rate for all of the organisms that live in the intertidal. And we can't say that we expect change to happen even at the same time. So not at the same rate, not at the same time. But we are expecting most species are shifting toward the poles, and we expect that to be occurring in the intertidal. We're also expecting, there's a range in the intertidal from the upper intertidal to the lower, and we're expecting to see things shift up and down in that range. So there's like this little range of just the intertidal, and then there's this huge latitudinal range of moving poleward. So the distribution of intertidal species are changing, just again, not at the same rate and not at the same time. We expect that abundances are changing as food sources leave, as food sources shift up and down in the intertidal, and also as invasive species move in, or as organisms that are right now in the south move in their poleward trajectory. These aren't invasive species per se, they're just migrating northward. And along with that, we do expect that things change in their phenology, especially organisms that are on the southern part of their range, like Ascophyllum nodosum, the rockweed that we're all studying. And thank you so much, everybody who's contributing the data. This is a species that we're kind of closer to the southern end of the range than we are to the northern end. And that means that this end of the species range can be a little bit more vulnerable to change than, say, smack dab in the middle. So it's going to be it's an ecosystem of surprise, because we don't know how different species will respond to the changes of themselves and of the species that they live in the intertidal or visit the intertidal with. So we're expecting the unexpected when it comes to a changing climate and responses in the briny shallow. So Jesse, if you want to take it to rockweed?
Yeah, I can. So in terms of thinking about when we talk about rockweed, what are we talking about? So rockweed or rockweeds in general are a large group of, I would think, charismatic, amazing macroalgae or seaweeds that you find in the intertidal zone. But for this particular webinar, and when people typically talk about rockweed, they're really only talking about one particular species, which is Ascophyllum nodosum. And so it's highlighted here in kind of the yellow box. And so it's closely related to other rockweeds, which are in the fucus genus. So I've kind of highlighted those ones too, to see that when we say rockweed, It's sometimes specific to one species, but it's a large group of these brown seaweeds that occupy the intertidal shore and are really important in terms of the ecological health and well-being of rocky intertidal shores. And that all rockweeds are lithophytes, meaning that they need a hard substrate to grow on. You're not going to find them in kind of rocky or unconsolidated sediments. And in thinking about why they're amazing is that they are these long-lived perennial species that are foundational species that if you think about the coastline in New Hampshire and in Maine and in the Canadian Maritimes, when you have rocky intertidal shores, especially on sheltered embayments, it's rockweed that you're going to see and that you're going to have ever present. The width of the rockweed bed might change depending on the coastal topography, but it's a really dynamic, rich environment. These organisms are these foundational species, so they provide habitat, they provide shelter for all sorts of other organisms that most people who attend the shore are interested in. So they might move away the rockweed really quickly to look at a crab or a snail, but it's those rockweeds that are going to be maintaining that ecosystem for a really long time. And they're also primary producers. So in thinking about the importance of rockweed for coastal habitats, they're taking sunlight energy, they're converting it into chemical energy and sequestering that carbon as a long-term store. And they're also oxygenating the environment. So they're really important in terms of the health and well-being within any of those intertidal ecosystems. And thinking about rockweed itself in terms of its life history or its life cycle, it's a sexually reproductive organism, that there are separate male and female individuals. And this is the right time to go out and be able to identify the differences between males and females, that when they are reproductive, they have these reproductive organs called receptacles that are on the kind of the lateral fronds of any Ascophyllum. And that if you have a female, if you look at the receptacle, either kind of if it's actively reproducing or if you wanted to open up a receptacle, if it's olive green in the inside, you have a female. And if it's kind of this like tang orange color, it's a male. And that generally speaking, if you wanted to spend your afternoon looking at lots of different individuals, I can tell you that they're about 50% female and 50% male in terms of sex ratio in any particular given environment. But it's a great afternoon activity if you want to partake and validate it yourself. And in thinking about its reproduction and phenology over the year, they initiate their reproduction kind of in the middle, kind of end of fall in winter, and they become actively reproductive in terms of having these receptacles release eggs and sperm into the water column around now. So in terms of thinking about, depending on the temperature, May, April, May, and that usually they have finished reproducing in June, and they start to shed those receptacles. And so by July, there's typically no more receptacles left. They start vegetatively growing, and then they'll kind of go through that cycle again. But the relationship between sea surface temperature and reproduction is really tightly linked for Ascophyllum or for rockweed. And so this is why rockweed is a wonderful indicator species in thinking about the response of climate change to an intertidal organism. And so one can predict that there might be shifts in when reproduction takes place with shifts in the temperature. So as the water starts to warm, it's predicted that Ascophyllum might respond by changing when it's reproductive, and that it might become reproductive earlier in the year if it becomes warmer earlier in the year. And so a lot of research that Hannah and I have participated in and are actively participating in look at kind of this interface between rockweed reproduction, health, and understanding those rockweed ecosystems, both rockweed as an individual and then rockweed as an ecosystem, and that it's a single species that interacts with invertebrates and birds and all sorts of other organisms in the nearshore environment. And so one particular project that I participated in and was funded by the Maine Economic Improvement Fund was trying to understand just how important rockweed reproductive material, so all those eggs and sperm and fertilized eggs called zygotes, are to the nearshore environment. They could be important for filter feeders. They can be important for transient larval stages of economically important organisms, and really trying to understand who might be utilizing rockweed resources and energy in those systems. And so again, signs of the seasons data are really helpful in terms of understanding shifts in timing of reproduction, and then that cascade effect within trophic levels. So what primary consumers and secondary consumers might be relying on this resource, and how it might be shifting over time.
Sure. And so this is another effort that both Jesse and I have been involved with. This is the Maine Sea Grant funded Reconciling Multiple Stakeholders in Rockweed Habitats, also called the CRASH Project. And you can find out more about it on the UMaine website, or you can Google CRASH with two S's, CRASSH. But anyway, this is a project that we've been working on with Amanda Clemmer and Brian Olson at the University of Maine and other UMaine graduate students. And as Jesse mentioned earlier, you know, rockweed is an individual organism. It also creates an ecosystem in and of itself. And this research has focused not only on rockweed recovery in terms of biomass and other characteristics after a harvest of commercial scale, but also we've been investigating how the entire ecosystem responds to a harvest. And so we have been looking at how does the ecosystem in and of itself, in terms of temperature and light and wave action, how do those change with and without harvest, as well as what happens to the invertebrates that call rockweed home, and coastal birds who come and go and eat those invertebrates and you know, loaf around and that sort of thing. Also in the rockweed habitat. Loaf is, by the way, a real word for marine birds. You can look it up, Eider's loaf. So I didn't want to make them sound like lazy slackers. They're not. But anyway, so that's work that's ongoing now. All of the graduate students are busily working on their data to come to understand you know, the rockweed ecosystem in and of itself, and how the rockweed ecosystem responds to harvest.
Yeah, and so sort of branching off of that, of understanding, you know, how much rockweed is there, and are there better ways to be able to get a handle on rockweed health and monitor it in terms of abundance and distribution over time and space. Hannah and I and some colleagues from a private company called Nearview and from the Bigelow Labs for Ocean Sciences are using and trying to develop unmanned aerial survey systems, or drones, to be able to have coast-wide abilities to map and to classify, so distinguish the difference between Ascophyllum and some of their close relatives in the fucus, and be able to estimate biomass. So this research has been able to kind of take to the skies to be able to figure out how much rockweed there is, and kind of understand that distribution and abundance of rockweed versus some other similar related species. And so what we're doing there is developing methods to be able to remotely detect how much, first be able to figure out, can you tell the difference between rockweed and fucus? And then be able to start to get some algorithms to figure out, can we accurately measure rockweed biomass? And so these particular figures, I think, are kind of exciting in thinking about what the power is for some of those technologies that are kind of coming online. And so if you look on the left-hand side, you see all these little green squares or red diamonds, and those are kind of boots on the ground field groups that Hannah has led from Schoodic Institute, kind of measuring how much rockweed there is in a particularly small quadrat, and compared to what the drone might be seeing, which is a really much larger space over time. And on the right-hand side, on that upper panel, is sort of, again, the overlay between the quadrats, and then also the drone being able to then classify fucus versus Ascophyllum. So kind of the darker green coloration is the distribution of Ascophyllum, and that lighter kind of citrine green yellow is fucus. And so this is going to be really helpful in terms of figuring out how much rockweed there is and how it might be changing over time. But I think technology only has so much power, in that we really are relying and so grateful for citizen scientists to help us, in that technology will not be able to distinguish phenophases of Ascophyllum, and really be able to let us know how individuals and individual rockweed beds might be changing over time. And so in thinking about the folks who are on this webinar, it's really so important, the work that you're doing in terms of collecting those data and being someone who can distinguish the difference between different parts of the phenophases and being able to record it, and going out there routinely on all different parts of the coastline. So, you know, a drone can only go so far, a human being can only go so far, but a team of citizen scientists can really do so much. And so rockweed has been one of the indicator species for Maine Signs of the Seasons since it began. So this is a citizen science program that Esperanza and Beth created, and that Carrie is also now co-managing, and that is in coordination with the University of New Hampshire Sea Grant and the Coastal Research Volunteers. And that rockweed is really the only briny shallow coastal indicator species. So again, in terms of thinking about how little we know about how the marine environment, and especially intertidal environments, might change over time, it's so awesome that we have this one indicator species to be able to get a handle of how a particular organism might be shifting its reproduction over time. And also, hopefully, Hannah and I have convinced you how important this organism is for intertidal ecosystems. So understanding rockweed gives us a lens of understanding how intertidal ecosystems might be changing and responding to changes in climate over time. So really super important, and the work that you all have done has been really valuable in terms of adding to a data set to be able to see if there are changes over time.
Thanks, Jessie. Yeah, I want to just quickly point out that I started out as one of Jessie's volunteers. I still am one, but I have somehow managed to work myself into a PhD program studying rockweed, all starting out from being a Signs of the Seasons volunteer. So Signs of the Seasons volunteers and coastal researchers, we start out a little bit curious, and some of us, that's perfect. We stay at that little level of curiosity, and thank you so much for contributing your data. But what we've also found is that there are many, many different opportunities for folks who want to learn more, want to contribute more, that there are opportunities for people to expand the work that they do in the rockweed ecosystem, and there's a variety of things to be done. And so far, all of the folks who have come out and worked with us on different research projects, even if it's been freezing cold or blisteringly warm, there's no shade in the intertidal, by the way, or very little, you know, that their knowledge of rockweed and the interest in learning more and more and more about this fascinating system has been wonderful to work with, and has definitely pushed me, and I would probably guess push Jessie as researchers to really think differently about the system in which we work. So again, you know, thank you to everybody. And I did want to point out, there is another citizen science effort underway, going back to Jessie's question a few minutes ago about like how much rockweed is there. We do have another project, Project ASCO, short for Ascophyllum, but also for assessing seaweed via community observations. This is you know, a data collection effort that's occurring along the coast In Maine. I haven't really considered it going to New Hampshire, but I'm sure that it could. And we are working with citizen scientists to be those boots on the ground to collect data on strictly on the rockweed biomass along the whole coast so that we can understand better how much rockweed is there. Those data can be used by people who are using drones to assess at a larger scale. But like Jesse pointed out earlier, sometimes you just need boots on the ground. And citizen scientists have proven more than capable of really contributing deep data to this effort as well as other efforts. So we're exceptionally grateful to people who are contributing and who will contribute, continue to contribute. We started this project last year and it'll continue at least for another 2 years. So it's another good project for people who want to be involved with rockweed.
Cool. So, so then we're getting to like what are the data that we have so far? And I want to sort of couch kind of what I will present in terms of thinking about data, in that phenology data and climate change data are always really challenging. So when we want to talk about climate change, we're really talking about long-term data sets. When we think about climate, it's really long-term. And those long-term data sets, if you think about them, and thinking about all of the different types of data that have been collected over long periods of time, are really rare. I mean, they're really needed to be able to figure out, are there changes over time? But for lots of data sets, they're just beginning. And I want to kind of couch the information I'm going to be sharing with you is in the just beginning stage. So we may not be able to see patterns in terms of responses of climate. We might be just looking at patterns and responses to changes in weather. And so Hannah had elucidated this to me, and I think it's a really great analogy in terms of thinking about variability that might be annual variability that would be more related to, let's say, weather than to climate. So when we're talking about changes in weather, it could be like you look outside in a shorter term and you're saying, what should I put on today to meet the elements? So it might be like it's hot or warm and you choose a sweater or you choose a t-shirt. And that would be sort of like small scale variability that we'll put in the kind of like short-term weather, versus climate, which is not thinking about what you should put on today, but what your wardrobe looks like. Like, should you be investing in no t-shirts at all and only sweaters, or just muck boots and not flip-flops anymore in terms of the amount of precipitation? And so just trying to kind of couch what I'm going to present to allow you to see that we're just starting off in terms of gaining enough data and hopefully invigorating you to continue to collect data so that we can start seeing long-term changes and being able to detect climate responses as opposed to being able to characterize annual variability that might just be more like weather. So in thinking about the rockweed data that has been collected since 2014, most of those data are now residing in an amazing database called Data. And this database allows any user to be able to download the data. So if you're like, I want to know more about all of the data, the Signs of the Seasons phenology data for rockweed is there, but there's lots of other data sets that you can explore too. And since 2014, there's over 1,200 observations that you all have contributed. And so that's amazing. That is way more observations than Hannah and I could ever make in that period of time. And so I think, again, it just really highlights that we can't do this without citizen scientists. And we are so grateful to be able to get the breadth and depth of these observations, because we couldn't do it alone. And so in thinking about the data that are there, it has a really large geographic range. So the southernmost data observations are in Rockingham County in New Hampshire, and the most northern and easternly data sets that we have are from Eastport and Washington County, Maine. So we really have a really nice capture of the coastline. And in thinking about kind of how to look at those data, There are some observers who might not continue after 1 year of collecting. And so in terms of trying to look at the data as holistically and robustly as possible, what I ended up doing is pulling all those 1,200 observations and then clustering them together by county. So if we look at New Hampshire and Maine, you can see the stars on this map that kind of show where those data are in each of the counties. And so again, it's got a really nice geographic coverage, but not every single county is represented. So if there are folks who live in Saginaw County or live in Knox County who want to start observing and recording, I think those are data gaps. But again, in terms of just thinking about the really nice span of having volunteers and citizen scientists who are making observations as far down east as we possibly can have, and in terms of representing New Hampshire, we're doing a really great job. So really, thank you. And so what I'll kind of show you is what data we have so far for all of those different counties for all the years that we have those data. And they're incomplete, but again, in thinking about looking at data over time, we're just starting to collect enough data to then be able to look for trends. And I might have said this before, but I'll kind of hone in on it, is that phenology data is really messy. So it's really hard to just try to figure out what to show and how to show it. So this might not be the only way to display the data, but I'm going to show you the way I displayed it for today. And so this is kind of to orient you. Each of the years that I have for each of the counties that you've collected and input into ANIC data. And on the top of each of these little panels, there are a whole bunch of letters that represent April, May, June, July, August, September, and October. So those are the months of the year that most folks were out on the coast making observations. And sometimes there might be a bar that kind of extends out from April, and that would be any coastal observer or Signs of the Seasons observer that was going out earlier, but I didn't kind of extend out that range because it was inconsistent for everybody. But and then the other kind of part to orient you in terms of this information is the 4 categories that citizen scientists are inputting in terms of thinking about the phenology. So if there are new side branches that have been recorded, if it's reproductive in terms of having those receptacles, and then the phenophases of either smooth inflated receptacles or dotted inflated receptacles, or that the kind of the detection that reproduction has ceased, so having torn receptacles in terms of kind of shedding off those receptacles and starting vegetative growth. And so this is for Rockingham and Stratford County. In 2018, there was just 1 county and not both counties represented. And what you can see is that they're sort of all over the place in terms of having a lot of annual variability. So that there really doesn't seem to be a trend of certain phenophases trending earlier in the year or later in the year. I haven't kind of integrated temperature into this either, but just kind of looking at kind of overall trends, that there's just a lot of variability that we're just starting to capture and have these data points as reference for as we move forward in the future of what might be similar or different in different years. This is for York and Cumberland County. And again, depending on the observers that are able to go out and collect information, some years have better representation than others. And so again, there's just a lot of annual variability in terms of thinking about York County on the left and Cumberland County on the right, and that some durations of phenophases seem to be longer or shorter, but again, there doesn't seem to be a lot of kind of definitive, like the data are showing something quite yet. But again, really, really helpful that we have these data. That's I think the most important thing to kind of take away is that we have these data so that as we start to, again, accumulate more years to then refer back to, we have something of substance and of validity to look at. And thinking about Lincoln and Waldo County, although Lincoln County was represented for 2015, it is also a data gap in terms of thinking about that information. And again, a lot of annual variability between those different phenophases over time. And then our last two counties in terms of Hancock County and Washington County. And again, Washington County is a little bit data poor compared to Hancock County. But again, I think the takeaway that I had when I pulled these data and started to kind of plot them up was that there's a lot of annual variability, and we're capturing that, which is really important. There was only really 1 year. So in terms of just thinking about like, okay, I've got all this information, I'm looking at it from county to county, but I'm not really kind of looking at comparing all the counties in 1 year together. And so in thinking about kind of comparing geographically, are locations different than each other? 2015 was the year that we had the most comprehensive number of counties that had enough data to really compare to. And so if we look at this, this is from kind of on the top part of the panel is Southern Maine. As we kind of move down the panels, they kind of move more to the north and the east of Maine. And I can't really see any regional trends, but we have this information in terms of being able to kind of see that there are probably kind of a patchwork quilt analogy in terms of responses of when reproduction is occurring, that different counties might have different exposures and different embayments and maybe different physical or chemical characteristics that also might play a big role in terms of when rockweed is reproductive and the timing of its reproduction and the duration of different phases. So again, really helpful to have this information as baseline data and as reference data as we move forward to then compare future data sets too. So hopefully that's been helpful in terms of being able to look at some of those data as you kind of are just putting your numbers into the internet and then hoping that it goes somewhere, is that they are coming somewhere and that they're being kind of connected and then they're being explored to be able to see if there are going to be changes over time. So with that, we wanted to leave a bit of time for questions. We are so grateful for your for your attention for the last 45 minutes. But please feel free to ask any questions or provide any comments, or if you want to share any stories out in the field, please do so.
Thank you so much, Jesse and Hannah, for your wonderful presentations and for taking the time both to analyze these data and for doing all the other research that you're doing to help us understand more about our rockweed dominated intertidal ecosystems and the value of those systems and how they're interacting with other species. So I just want to add to what Jesse shared about and Hannah shared about the value of these observations. And I put in the chat a link to our volunteer interest form. We typically do trainings more in the spring, but throughout the year. And I know that we have a number of folks who have currently shared with us that they're interested in doing another coastal rockweed monitoring training. And so I'm collecting that information and would like to set up a few. So if you're interested too, please feel free to fill out the form or reach out to me. And we'll follow up with contact. I'll put my contact information in the chat in a moment. And Wells, did you want to offer just a summary, or Allison, of when you'll be doing your trainings in New Hampshire as well?
Sure. Yeah. For the coastal research volunteers, we'll be doing our trainings typically around late January, early February next year.
Great. Thank you.
Thank you.
And so I would love to invite any questions, and I'll just start with one that Susan entered into the chat a few minutes ago. You write, Susan, one of the most important season signs for us at Source is spore sac drop off. And you're asking about when that typically occurs. Dr. Bob Vadas had shared that it typically occurs when the water, according to water temperature, when it reaches around 6 degrees Celsius. And the question is whether Jesse or Hannah, you have any information or further information about that, the importance of that timing?
Hannah, do you want me to take it? Okay, I would say great question, Susan. And I would, I think that Susan's question is coming from someone who utilizes the resource too, in terms of really important in terms of thinking about waiting for reproduction to cease before harvesting rockweed. And that's really a very helpful best practice. I would say that, been looking when in 2013, when I was kind of doing some more of the subsidy work, trying to go back and see if the Bacon and Vadas data kind of still held, since it was in the 90s that they were kind of collecting those data and kind of making those predictions, and it does. So in terms of just thinking about the onset and then the peak and the termination of when reproduction occurs, it is temperature related. I think I can't remember off the top of my head if it's 6 degrees Celsius completely or not, but it's around that range in terms of thinking about as the water really starts to warm, reproduction is going to cease when it's really, really, really hot. So we see that in terms of, again, that shift in phenology, that there's a lot of annual variability. And 2012, it was a really, really warm summer and a really early warm spring. And Ascophyllum reproduction was really early that year. And same in terms of ceasing when those receptacles were shed. So it still is probably the best environmental variable to be able to predict when those receptacles are going to be shed off of the Ascophyllum.
Thank you. And there another question is with a question from Morgan. With that period that you referred to of extreme heat, was there any specific impact on Ascophyllum that was observed during that time?
Thanks, Morgan, for the question. So that period of extreme heat that I was talking about occurred in the Pacific Northwest, and Ascophyllum nodosum is a North Atlantic species. There are rockweeds that they're just called generically rockweeds. They're not the same species. There are rockweeds that occur in the Pacific Northwest, and there was some impact, but mostly on the rocky intertidal in the Pacific Northwest. It's shellfish or barnacles, and then it's some kelps that are sort of in the lower intertidal. There was high mortality of mussels, of barnacles, and of some of those kelps that were higher in the low intertidal. There's not massive rockweed domination in the areas where they saw high death. So there was some death to some rockweeds, but because Ascophyllum is a North Atlantic species, it was far outside of the impact zone. And I hope that answers your question.
Yeah, I mean, I would say that rockweed, if you go out, I mean, it's all of these perennial species are incredible, and that if you think about the coldest cold days in the winter and the hottest hot days in the summer, rockweed persists. It's a really resilient species. It may lose some photosynthetic pigments in terms of kind of bleaching and becoming a little bit more yellow. That might be a stress response. It also could be that vegetatively it's just growing a lot faster and it hasn't kept up its pigment concentration as it's growing. But it's pretty incredible, I think, in terms of if you're paying attention to rockweed across the calendar year, just how extreme those temperature ranges can be, and that they persist and they do incredibly well.
Thank you both. So there's 2 more comments, and I'll just add that if you have a question, and you can either put it in the chat or you can use the reactions feature at the bottom of your screen in Zoom. If you click it, you'll see a raise hand feature, and I can certainly call on you. Morgan had a follow-up, which was, do you see an increase of nutrient inputs when the receptacles are released?
That's a big question, right? So I think my prediction is that when you have so many eggs and so many sperm and so many fertilized eggs being pulsed into the environment that they are a massive uncharacterized carbon and nitrogen input into the coastal systems. We haven't totally characterized that yet, but we do know that organisms take up, especially fertilized eggs, those zygotes, and they retain them in their tissue. And so we know that organisms are utilizing it, and that it's not around for a really long period of time, but it's really important probably for lots of organisms that are relying on that seasonal source and pulse of carbon and nitrogen into the environment. Those sperm are also bright orange, and that is a carotenoid pigment. So in terms of just thinking about folks wanting to have antioxidants in their diet, that's a really great source of antioxidants. So in thinking about the high stress of intertidal systems, if you have a filter feeder that's being able to consume a high antioxidant source of nutrition, like surplus sperm, that might allow them to have that additional nutrition in periods of when they might need it too.
Thank you. And Susan, you also had a follow-up about the challenges of collecting water temperature data at different depths and trying to understand an average temperature for a whole stand of rockweed, for which you're dealing with tidal cycles, and which changes the temperature dramatically also. So I don't know if you wanted to follow up on that, but if Jesse or Hannah, you want to, feel free to jump in, Susan, but if Jesse or Hannah, you just want to speak from your own experience about that challenge and how you've managed it, that'd be great.
Susan's right. Right. I think in terms of being a coastal research volunteer or Signs of the Seasons volunteer, it's just critically important that every time we go out, we measure the temperature at the same location each and every time we go out, so that we can all minimize the variability of where we're getting our temperature reading from. I mean, that's what I take away, you know, always being super duper consistent, just like we're always very consistent with where we get our phenophase data from, getting our temperature data from the same place every single time as volunteers is critically important.
Thanks, Hannah. And so there's another question: with temperatures getting warmer earlier, the window for spore production could be shortened. Have you seen any indication of this yet? And secondly, when are spores ripe enough at the earliest to fertilize or release productive eggs?
Yeah, so I think, I mean, that really is part of where Signs of the Seasons is so important for these data. So in thinking about that duration of when reproduction occurs, I think it's going to be different at different locations. That, you know, we can't assume that all rockweed is going to respond like a blanket similarly across a geographic distance from New Hampshire to Maine or to the Canadian Maritimes. And so they might be responding differently in that maybe New Hampshire has a shortened reproductive period of time and down East Maine doesn't. We don't know that yet. So in terms of thinking about that, I think we're in that discovery phase of figuring that out. And every year is going to be different in terms of just thinking about how cold the winter is and how, like, when the start of spring occurs. Is it just a blip? You know, like there was one day in March that it was like 70 degrees and then it plummeted back down to 40 degrees. Does that trigger holistically reproduction? So I think that's where the real value of citizen science data to be able to start to answer those questions occurs, and I can't answer that right now. But one would predict that with a warming Gulf of Maine, we're kind of extending out our summers, and that we are probably truncating how much kind of cool periods there are. And so that we might be seeing over a longer period of time a real shift in starting of reproduction earlier. And again, those data are showing a lot of annual variability and we're not seeing trends yet, but that's what one might predict.
Thank you. There's one more question. I think we have 1 more minute, so we'll give it a shot. This is a question from George. Is the importance of the reproductive cycle to maintain the density of the beds, or as a food source for other intertidal inhabitants? Or maybe the answer is both. Can you respond to that?
I'm going to go with C, both. You know, I mean, we've already said that the intertidal is a crazy place to live and to try to latch on to grow. And so the cycle, not so much per se the cycle, the fact that there is this high reproduction is critically important for maintaining the density of a rockweed bed, because we need to have the future generations always in the understory growing up. In terms of the cycle, that's kind of critically important as this pulse of food that comes into the system in the springtime, especially when there are a lot of organisms that are having their young or releasing their young at that point. So it's a critically important food source as well. So both, George and Jesse, do you want to add to that?
Right. I think it just sort of highlights that kind of duality that rockweed plays. It's both an individual and it's part of the ecosystem. And so in thinking about it, reproduction is really important for an individual and perpetuating that genetic line over time and that species over time. And it's also really important in terms of being part of an ecosystem that relies, I would say, on surplus reproductive material. So it's not that these organisms are like consuming all of those eggs and sperm. There's just trillions of it. I mean, there's so much reproductive material that there's more than enough to perpetuate the species and also be able to feed that nearshore community. So I like C as well.
Thank you. And I'm sorry, but I think we've run out of time. Thank you for your great questions. Thank you for your wonderful presentations. I saw that, Wells, you put in a link for the Coastal Research Volunteers website as well. And I've just added my email address if any of you would like to follow up with additional questions. If I don't have the answer, I will connect you with whomever else on this webinar can answer it for you. And I just want to thank you all so much on behalf of Maine Sea Grant, the University of Maine Cooperative Extension, and also, if I may be so bold, on behalf of the New Hampshire Sea Grant and Cooperative Extension and the Schoodic Institute and Maine Maritime Academy, for whom these data have been really helpful for research endeavors. So with that, I think we'll end our program today, and we hope that if you haven't yet had a chance to volunteer, you'll get involved and keep making your observations if you're already engaged in our programs. Have a great day. Thank you so much.